Okay, I think it's good time to start. Good afternoon, everyone. Welcome to the final report of ICBS. I'm very honored to introduce Professor Sun Fengzhu from University of Southern California. He is an expert in the area of computational biology and bioinformatics. And today he will give us a report of statistical and machine learning approaches for investigating virus host interactions. Let's welcome Professor Sun Fengzhu.
Thanks for the introduction, and thank you for staying late into the last lecture, and thank you for attending my lecture too. So today I'm going to talk about our recent work on statistical and machine learning approaches for investigating virus host interactions. Now, this topic is related to metagenomics. So I will give a brief introduction about what is metagenomics. Then I will talk about a computational method for classification of metagenomic contigs. And so contigs, you can think about long sequences, and I will see why we call it contigs. Then I will talk about the method for matching virus and their host. And finally, I will talk about how do we identify virus-host interactions in metagenomic data. In particular, how do we analyze metagenomic IC? So what is metagenomics? Metagenomics is the study of genetic materials derived from the environment, such as from the human body, from the human gut, or from your nose, from mouth, etc. And the human microbiome have been shown to be associated with several complex diseases such as inflammatory bowel disease, colorectal cancer, because those are all related to the human gut, and a lot of microbes there, and that's understandable. And the other things that related to like mouse, and so in our mouse we have lots of microbes, and so that's another area that the microbe play a very important role. And some have been shown that the microbe can also impact the response to certain drugs, because the drugs depend on the metabolism, and so that's definitely the human gut. Microbes just generate those, metabolize those things, and it's just to give you some, how does the person respond to the drug. Not only human health, and the other environments such as from the ocean and as well as soil, and that's also very, very important, because we have pollutions and we have also climate change and so on. So that will change the microbial community in the ocean as well as in the soil. And so how does that will impact the healthy of those healthy of the ocean as well as the soil are very, very important problems. So therefore, the microbiome studies play a very important role from the human to various environments. About 20 years ago, Craig Venter, I think many of you know, he is just sequenced the human genome. After he sequenced the human genome, he went to the ocean and did the global ocean survey. And that started the microbiome project. And so he used those sequencing technologies to survey all those different environments. After that, then there is the human microbiome project to try to study the microbiome in various parts of the human body. And then there is the Europe MetHeat project, study the microbiome in the intestinal tract and how does that relate to the disease. And then there is the Earth Microbiome Project and the National Microbiome Initiative that's more related to the earth, to the soil, as well as to the ocean. most recently, the human viral project will be started this fall. So this you can see from this all this international as well as U.S. microbiome project, and see the importance of microbiome studies. So because my main collaborator is a marine biologist, I just use the water as an example to show why we want to study viruses. So in a drop of water, we have about 10 million viruses, 1 million bacteria, and 1,000 protists, etc. Although most of the genetic material come from bacteria, because bacteria has much longer genomes, and so about 90% of the genetic material you sequence from the microbiome are bacteria, but the number of viruses are much larger. As you can see, the number of viruses is 10 times of the number of bacteria. In addition, you can see from the recent COVID-19, the small virus, although it is quite small, it can cause very big effect. It just changes the whole world. Similarly, in the ocean, the viruses affect those bacteria can play a very important role, and it can change the dynamics of the ocean and the other environment. So therefore, studying viruses are quite important. However, because the number of the genetic material from those microbiome are mostly bacteria, so the studies of viruses lag behind. Another thing that is related is plasmid. Plasmid also affect the host and impact the survival of their host. So in our recent studies, we try to study the so-called mobile genetic element, including viruses and the plasmid. The story of our studies for the viruses come from our collaboration with the biology collaborator Jed Furman, who is an expert on viruses. He was the first to discover the viruses in the ocean in the 1990s. So he used some biological techniques such as filtering. He knows most of these sequences should come from viruses. But when he mapped the read that he gets from those experiments, only 10% of this read can be mapped to the current databases. His question at that time was, Fengzhu, can you say if those sequences are viruses? He said, I'm sure that most of these sequences are viruses, but I can only map 10 of them to the ACBI. So the reason is because our studies for those viruses are very few, so only very small fraction of those viruses are in the current database. So now the question becomes, for a given sequence, we ask the question, is this sequence from the viruses or not? So that's become a computational problem, or you can think about a machine learning problem. So here is a kind of conceptual formulation of our study. And so here we just make those, like here, these are in the environment sample. And you can think about, we have a variety of different microbes, and they are including those prokaryote and the eukaryote, and their genome sizes are much longer. And but we don't know them, okay? And so for those sequences, those are viruses or plasmid, they are much shorter. And these colors in indicate the different type of microbes. So those are unknown, and we only know are just a drop of water or just some soil, and then we get them, and then we don't know what are there. So how do we say what are there? And we do sequencing. So they just do random sequencing, and now after we do sequencing, and then what we get are those short read. Those short read about 200 to 400 base pair long. So we don't know where they are from, and so we don't know their characteristic. And so all we know are the ACGT of those base pairs, and then those are the data we have. So we start from the computational biologists, come from those here. We have all this, and although I indicate all them as the colors, but we really don't know what color, and all of them are just sequences. Okay, so how do we study that? So the first thing we do is to sequence assembly. And so what it means is now suppose you have two sequences that they overlap, and then we just assemble them into longer sequences. But it's in reality that is too slow, and there are some other more advanced computational method to do assembly. So this is still an active research area, but I'm not going to talk about this because I did not really work on this particular problem. So we use other people's program, we do assembly, and then we get longer sequences. And so those sequences should just connect to each other, and they come from the consecutive regions of the same genome. And so we call those the contigs. So that's the first concept, it's contig. So those are the contigs, and this can be 1,000 base pair long, and some of them can be tens of thousands or hundreds of thousands base pair long. So those are the starting point for our study. Okay, so from here, and each of these contig are just sequences, ACGT, and so on. And so now the question is, what we start from here, we want to ask, what are these sequences are? So it's a sequence classification problem. We try to use from these sequences, we try to classify them. And that's purely from the sequence data. Okay? And so from here, we classify the sequences, and then after we classify these contigs, and then we study the abundance of the different contigs, or if we do binning, and we study the abundance of the different so-called operational taxonomy units, or you can call it some microbes, what we call it microbe, but it's not really. And then we study the abundance, and then we have some of these mobile genetic elements. And once we identify them, and then we also try to link those the prokaryote eukaryotes with those mobile genetic elements. And so who is linked with who can affect them. So after that, finally what we try to do is try to associate the abundance or the diversity of all those microbes, as well as those interactions with the function of the microbial community or human health. So that's the whole pipeline for analyze microbiome. Okay, so now in this talk, I will concentrate on the computational method to classify contigs into different classes, including prokaryote, eukaryote, viruses, and plasmid. And we also want to talk about how do we predict mobile genetic elements and host interactions. Okay, so I will mainly talk about a recent publication on the classification of metagenomic context that was recently published a few weeks ago. So, okay, so these are the I know some of you are college students or high school students, and so I will give you a brief history of what we did. And so, actually, as I said, we started this project about 8 or 9 years ago. And so the first is at that time, and we don't have deep learning. And so now it's just a classification problem. And so, so the input is the sequence data. So, I mean, it's in most of these machine learning tools, and you are just, generally we have the statistics, we have the x1, x2, xn, and then we input them, we just do classification like random forest or supporting vector machines to do the analysis. But here, what we have are the sequences. And at that time, how do you change those sequences into the so-called x1, x2, xp? And so, so at that time, what we did is to try to use the so-called camera frequencies. And the so-called camera is a consecutive sequence of K letters. Like if we have talk about formats, we might consider GGCT as one former, and we consider all those camera frequencies. And we use the camera frequencies of a sequence as those features. And then, so these are the features, and then we use the relatively simple penalized linear regression to do prediction for those viral sequences. So, although this method at that time is not that advanced, but it's solve a very important biological problem. Actually, it's because this is the first time using camera frequencies and using machine learning approaches to identify viral sequences, people get very excited at that time. And so Nature News recognize the importance, and then they report this work in the Nature News, and then that gets a lot of attention for that. And then 3 years later, and we have the deep learning, and we extend this work to the deep learning, and we also extended the database, the training data. And so those two studies play a very, very important role in the field of phage or the virus studies in the microbes. And so this, in particular for this deep viral offender, it had been used widely in the development of phage databases. And I said that phage databases are quite inadequate, incomplete, and so we need to develop phage databases. And so many groups used this tool and another tool called viral offender and to build databases. So those are the two major tools for the identification of viruses and to build phage databases. So in our most recent work, we extend this work to the classification of sequences into multiple classes. And so because this is a kind of mainly mathematical conference, so I will give you some outline how we did it, and those are the computational things. And so one is to build those deep learning model, we need to collect the data. So that's what we did. And so we collect a variety of different data. And so we have collected the prokaryote data, we have collected the eukaryote data, and well plasmid. For viruses, we collect those prokaryote viruses as well as eukaryote viruses. So those are the 5 major classes of those sequences. So when we do deep learning, we need the training data as well as to evaluate it. We use testing data. For the training data, what we used is try to simulate the real-time scenario. Suppose, although we did this study around 2022, but we set up the January 2020 as the time that study, and because sequences tell you which time it was discovered, and then you use that as training data, and also the validation data, and then you use the data from before the 2020 as the training data, and then you get the model. And then we test the model with the data that after those 2020. And in particular, we also restrict those training data to be not too close to the training data so that there are no information leakage. Okay? So that's just the collecting data. That's now for me to say this is easier, but it takes a lot of time for my students to really do it. So for the deep convolutional neural network, and so the input sequence is just the sequence, and then we have the forward string, and so for each string we consider two types of encoding. One is the nucleotide encoding. Essentially, we just consider the ACGT as just the token, and then sure we need the camera, decide what camera we should use. And then we also used the so-called nucleotide encoding, amino acid encoding. And so that's just the reading frame, that's 3 letters, and because when we predict those viral sequences are not, and some protein information can be very important. So we used those amino acid encoding too. The framework is not that complicated, and so we just have 3 layers of network, and for each of them, and then we do this. We just do this one, and then we get the embedding, and similarly we get the embedding for the reverse string, and then we connect them, and finally we just use the linear layer to predict them into the 5 different classes. So that's the design of those deep convolutional neural network. Okay, so what are the performance? And so here it just shows the performance. And here you can, the figure, if you just look at the left panel, and then this shows the AUC curve for different sequences lengths. And so these are the sequence lengths for 500. Those are the 1,000, 2,000, 3,000. And you can imagine if your method is readable, and as the sequence length increases, you have more information. Your prediction should be more accurate. So we try to study how the prediction accuracy increases with the sequence length. And so that's how we did this. And so as you can see, all those AUC curve is greater than 0.9, and also the accuracy increases with the sequence length. But there are some problems, and when we look at it carefully, such as for this one, you can see for the plasmid, the AUC score is 0.9. For the precursor, the AUC score is 0.92. And similarly, you can see for each of these, the plasmid and the precursor, that's give you lower prediction accuracy, although it still increases with the sequence length. The reason for that actually is there are some similarity between the plasmid and the precursor. So it's hard to distinguish them. And so when we try to merge these two sequences into one, like plasmid and the prokaryote into one, and this increased then we have a 4-class classifier, and you can see that the prediction accuracy are increased, and like we merge them into just prokaryote, and this from 0.99 to 0.942, and now it's changed to 0.94. And similarly for this one, it's from 0.92 to 0.94 to 0.95. So now you can, for 4-class classification, it become more accurate. Okay, so the other thing that we validated is through some simulated data that we have simulated various environments with different combination of prokaryote, eukaryote viruses, and plasmid, and we put them we have 20 different scenarios. And I'm not going to give you the detail of these 20, just to show you it's quite complete. The reason for we doing this simulation is in the real simulation, in the real data, you don't know what are the combination of those the different microbes. So then some experiment can also change the combination. Like you do filtering, you can filter out those eukaryote, you can filter out the bacteria, although you cannot completely filter them, but you can change the combination. So therefore, what we try to do is try to study the accuracy for and the different environment. So for each panel, for this panel, those are the prediction accuracy for the 5 different classes. And we compare with a variety of different software out there just for the virus identification problem. And this is the first algorithm that classifies those sequences into the 5 different classes. So we here just talk about the virus identification. And so we compared with other programs that do the virus identification, including our own program called the Deep Viral Finder. And then we showed that our method performs the best. And so these are the F1 score, those are the AUC curve. And for the eukaryote viruses, only our program and the virus sorter can predict eukaryote viruses. And then our performance is slightly better than the virus sorter 2 for those scenarios. Okay, so we also apply this program to some real data that our collaborators have been collected for over 20 years, and we get some important findings that at least my collaborator are happy about. Okay, so that is the first part of how do we identify the viruses and how do we classify those contigs into multiple classes. Before I go to the next topic, any questions here? Okay. So, once we identify those viral sequences, the next question is, how do we identify their host? And so this is an important problem in terms of understand the dynamics of the microbiome. And another important thing is the so-called phage therapy. So what is the phage therapy? And the phage therapy is trying to use phages to kill some viruses, the pathogens. And the usual application of that is antibiotics. When we get some bacterial infection, and then we use antibiotics. But because of the wide use of antibiotics, then there are a lot of antimicrobial, the so the antibiotics is not just effective anymore. So that's just antimicrobial resistance genes. And then we need some other approaches to kill those pathogens. Okay, so because viruses, you can kill some bacteria. So they try to identify those viruses that impact or kill some bacteria become a kind of hot topic these days. So, okay, so that's just one particular application for the virus-host interaction. So, what information can we use to predict viral-host interaction? So now it's a little bit biology again. And so the first thing is what will happen if a virus impacts bacteria. So we know it's that when we caught COVID and we all get the immunity mechanism, right? And so the bacteria actually are smart as human. And the virus affect some bacteria. The bacteria is smart enough to get some viruses from those bacteria, from the virus, and they get it into their own genome and in some repeat readings, and it's so-called CRISPR. And the CRISPR actually we know the CRISPR is a very important technology right now for the gene editing. But actually the first discovery is through the microbiome. So they get this, and so now you can see is if the virus and some bacteria share some CRISPR information, it indicates there are some interactions between them. Okay, so that's some very important information. If you see this, you are pretty confident that those viruses affect those bacteria. So that's one. The other information is the so-called alignment. So the alignment between the host genome and the virus. So what does it mean? It's when you do some local sequence alignment between the virus and the host. Some alignment between them, that indicates some interaction. The reason is they live in the same environment. There are some horizontal gene transfer between them. They are more easy to change genetic material. So if there are some alignment between them, then that's indicated interaction. So that's both of them have some sharing information there. Okay, so another thing is the so-called alignment-free method. Actually, our group had, before we studied this problem, my group have been developing measurement, alignment-free measurement for comparing two sequences and how best we compare the two sequences using the camera frequency. That's the alignment-free method. So we have been working on this for over 10 years before we studied this problem. So we used this measure, the so-called D2 star, that we kind of get recognition for this piece of work too, some time ago. And then we used this measure, D2 star, or the change to similarity measure, that's S2 star, to measure the similarity between the virus and the host. And again, it's because they live in the same environment, and so their camera frequencies tend to be similar. But we don't use the simple sequences between the camera frequencies, and then we use the so-called background adjusted camera frequency. What it means is you have the count of the camera frequency, you minus the so-called expected frequency. And the assumption, certain assumptions like Markov chain model. And so that's again getting to some basic the mathematics, and we use Markov chain to model the sequence, and then we calculate the expected count of certain chimeras, and then we can remove this background and then compare to those relative frequencies. Actually, if you go back to some statistics, that's related to the likelihood ratio test statistic, because the whole very important part in the statistic is to remove the background. And so that's the likelihood ratio. So anyway, so this is some earlier work, and then we showed, this is another publication, we showed that S2 can be used to measure the similarity between virus and the host, and can be used to predict virus-host interactions. So other method, after we publish our work, and then there's another group used the so-called WISH, and then they used the Markov chain model to measure the similarity between the virus and the host. So the idea is, again, there are some mathematics here, is what they do is the following. So for each host sequence, they built a Markov chain model. And then, once you have this Markov chain model, you calculate the probability of the viral sequence and this Markov chain model. And then you use that to measure the similarity between the virus and host. So again, you can think about this as the so-called the different host as a model, and you choose a model that should have the maximum likelihood. Okay, so from there you have this two alignment-free information. Another information that we can use to predict the virus-host interaction is the following. So similar viruses tend to infect similar bacteria, and the similar bacteria tend to be affected by similar host, by the similar virus. So that's just the biological fact. And so we talk about alignment-based method, alignment-free method, and also network method. And how do we make a unified framework that we can integrate all those information together to predict viral-host interactions? Okay, so that's getting, again, it's a computation gets a very beautiful mathematics here. And so now think about this. Now what we are trying to do is now for new virus, you try to see, okay, so which one I should link to the bacteria. And then what you do, you can think about this. Now, I think most of you know the recommending system. And in particular for the Netflix, famous Netflix problem, you try to have a new movie. Now you have a new movie come out, and then you try to recommend it to the customer, right? And so now if you think about this, and then you have the people, and then you have the movie, you try to do this. And it's not that much different from this kind of problem. So now you can think about this is a two-layer network. So one layer is on the top layer, we can consider them as bacteria. That's the bacterial network. And so for the bacteria, you can think about they have some similarity. And the similarity, again, you can use a variety of different similarity measures. And the most simple one, and the most that one that can be applied to any two sequences, that's what we use, is the S2 star, and so that's the method that we use. And then for this bottom layer, and that's the viruses. And then similarly, you can measure the similarity between the viruses using the S2 star. Okay, from there, and here, from the top and the bottom, that's the link the host and the viruses, and then you can also use them to link them. So now you have a two-layer network, and the top layer you have a similarity, bottom layer you have similarity, and then the top and both layers you have the similarity. And another type of information, you already know some of the interactions, some of the links between the top and the bottom, you know some of them should be linked. That's already in the database. Okay, so that's just become, you formulate this into a mathematical problem. Okay, so once you formulate it into this mathematical problem, and you do some mathematical manipulation, or you use what you learned using the recommending system, and you can try to see if you can get a good result or not. And not necessary, but you can try. Okay, so that's the link, that's the good thing, that when we do computational biology, and then we can link to some other things that we already studied in the machine learning or statistical community. Okay, so now I give you some mathematics, and so after this formulation, and then we can get some this formula. So the probability that the virus and the bacteria interact, that's the probability that VB equal to 1 that indicate interact, can be written as the following form. And so alpha is the background, and the beta, that's the S2 star, that's the indicate the virus and host similarity, that's here. And then the gamma plus is just indicate the similarity between this virus and all the viruses that affect those bacteria. So that's just how similar this virus is to the existing viruses that impact this bacteria. And this one is the SVV minus is how similar this V prime, this V is to those V prime. And so then this one measures similarity based on the CRISPR. This one is based on the alignment. Okay, so yeah, this you have this, and so yeah, this is a two-layer network, but this is actually you can consider this as once you have this, and sure, you don't know all the interactions, and you can use some Markov chain Monte Carlo method to do all this inference and so on. And so that's how it's mathematically become quite useful. And so actually this type of work is related to some work that we did in the early 2000s, about 20 years ago, on the protein function prediction. And that also it's become a kind of work that I was often cited for all my kind of honors. So anyway, so yeah, that's all linked to over 20 years ago. Okay, so I'm not going to give you the detail of how do we do the calculation. I just give you the work, how do we evaluate this work. And the first is we evaluate it. So you can see here, with this model, we can say, okay, so if we only use the alignment-free method, what will happen? So this is we use alignment-free. That's our first publication on this topic. And so if we use CRISPR, what will the result? If we only use alignment, what's the accuracy and so on? And if we use only the network information, so anyway, so essentially it's just you can see here that's at the genus level, and this is the family level, and so at the different levels. Yeah, it's sure it's at a very high level. You can predict more accurate. There's no that should be. And so, but if for each of these at each taxonomy level, and you can see it by integrating all this alignment free, as well as network information, you can get much higher prediction accuracy. Okay, so that's one. So the other one that we studied is how does the sequence length impact the prediction accuracy. And so here it's we know that for context, you may not have the complete genome, and how your results will change if you have just the context, not the whole genome. And so we can see here this is just the wish program. And so if you just use wish, and then it doesn't really change that much for the different sequence lengths. But when you use the integrative method, and we can see here, this is for 5,000 at a different, again, this is at a different taxonomy level. And so at 5,000, you have this one, and at the genus level, and when you have the 10K, it's getting this one, and when you have the 20K, and then you have those type of accuracy. So as the sequence length increases, again, you get more higher accuracy, and that's getting more accurate. So, and also the increase is not marginal. Okay, so that's another prediction for the virus-host interaction based on this network integrates a variety of different resources. And so again here what I see is the people just really see that if you use the integrative method and use it effectively, and generally it's give you good result. Okay, in the last 10 to 15 minutes, I will change a little bit topic, and here is in the topic that I just talked about. It's we only give the sequence information. So we did not really see in this particular sample whether there's the virus-host interaction. So here it's just, oh yeah, this virus had affected the other bacteria before. But it does not indicate that in this particular sample, and there are some interactions. Okay? And so how do we study if there are some real interaction happening in this particular sample? And so in this case, and we have to use some use one sample to see if there are some interactions. So this is not just a computational problem. There are also some experimental things that need to be carried out. So in order to do this, and the experimental technique to do this is so-called metagenomic high-C. So the high-C experiment is what the full name is. I cannot pronounce it now. And the high-C was originally used in the human, in one particular organism, and they tried to study how the different interactions and how the genomes formulated in the particular organism. In some cells, and they may change the different shape and where they are. So anyway, so high-C in a single organism is a very, very hot topic actually. And it's form a particular research group, and a lot of people are working on that particular problem. Okay. So metagenomic high-C is an adaptation of this high-C experiment in single organism in those microbiome. So that's simply say it's like that. So here I give you some background and how that was done. So what it does is just have the microbiome, and then the first thing they do is to use some biological enzyme, and in particular it's formaldehyde, and then they just link those genomic regions that are physically close together. Okay, so now those are the enzymes that are link those genomic regions that are similar together. Okay, those black ones are the linkage. And then they use some restriction enzymes to cut those linked genomes, to cut this linked genome. And then after that, they wash away those ones that are not linked together. And then they get them, and then those are linked together, then they ligate those ones, and they form them, and then they sequence all this short read. And so now you can see here, because they link those regions that are physically close together, but they may not be genetically, that on the genome, they may not be close together. They are just physically close together. And then, so those just form hybrid rate. And then they have the hybrid rate, and then they sequence them, and then that's the so-called the high C rate. And one end comes from this region, another end comes another region. And then so those are the metagenomic high C experiment, and from there they just say, okay, so how they link it together. So that's the metagenomic high C experiment. And so our computational pipeline is the following. So now you have the short read data. You still need the short read data. You have the short read data. You also have the high C read. And then you map those one end of the high C read into all the contigs, and another end to all the contigs. And from there you see where they are mapped. And then you can, they may map to different contigs. And then these contigs are physically close together. Okay, so now for the viruses, and then they have, if you, one of the contigs is virus, and then they might, this indicates that they are physically close together. Then you can link them. They are indicated. So if there are linkage between a viral contig and a bacterial contig, then that indicates the viral host interaction. So that's the basic idea. Conceptually, no problem, and that should be fine. And so, but is this true or not? So now what you see here is the relationship between the count, the number of links between two contigs, with the production of the product of the length, the number of restriction site, and the coverage. And then you can see there are some increasing trend there. Once you see this, it's just to say, okay, so if two sequences are, both contigs are long, more likely they are linked together. So if you just based purely on the number of connections between them, that's, you most likely you just say, okay, so they are, just because they are long, and it does not mean that they really interact. So therefore we need to normalize this data, and then that's become a data normalization problem. And so my students did some normalization and so on, and then we developed a method to analyze those high C data, and then to study how the viral host interaction in particular samples. So I think my time is up, and then I'm not going to give details on this this piece of work, but it's I just see it's this student is just amazing. I mean, he's within 5 years, and he's getting 2 Nature Communication papers, 1 Genobiology, and 2 Recom papers. And so immediately after PhD, he got a faculty position in the computer science department in University of Texas at San Antonio. So that's my first student. So immediately get a faculty position. So anyway, so I'm very proud of him, and I just say something that just to get some acknowledgement. So many of this work is just the early part of work is done by my former students Renjie, and she did the viral finder, deep viral finder, and also helped with the viral host matter. So she works in the DeepMind, the Google Brain. She's she worked on this and also worked on a lot of machine learning things, and she was selected as the top 50 Chinese AI researchers in 2002, and in 2003 selected as top 20 female researchers on March 8th, and so that's the Women's Day, right? So that's the early part, and for the Yishan Du is just did the metagenomic high-C work, and that's great to have so many great students that work on this. In terms of biology, you can see a lot of things that I need to collaborate with them, and so my collaborators including Dr. Shenmei Ho and also Jed Furman. That's my collaborator for over 20 years, and Mike Wortman was my PhD advisor, and he also quite involved in this project. Thanks for your attention.
Thank you very much for the very nice talk. Any questions from the audience? Okay.
Thanks for the wonderful talk. So I was wondering, like the context data or the sequence data is very much like the text data or the language. So like the methods you use, like a k-meris, also has a analogy in the text data. I wonder, there are some recent developments in language model. Like I don't know whether those techniques can contribute.
Excellent point. It's it's like a transformer and other things. The encoding. All these are just kind of like this text type of data. So how does all these things just related to this, and how if we use those things, how the performance. So this definitely is a very important. I know Professor Yan Yisun's group from Hong Kong, and so she actually put by ICAR paper there and used the large language model to analyze that. So she showed yes, it's the best. And I still need, and it's when we see people say the best, I need to wait until some independent studies. But I think it's very promising field. And how does all those transformer things just use those embedding? And the one thing that I look at, I think one thing he said, cameras is not the best thing to look at, but it's like the general language, you token this, just find the token first, and then do this embedding. It's better than just use the camera and the tokens.
Okay. Any questions or comments? Okay, I think the time is close to the closing ceremony. Yeah, let's thank you, Professor Sun again. Thank you.
Thanks everybody again for attending.
穹苍广而横千，物和多容。星云璀璨，黑洞冥冥。观测天象，探问宇宙，基础科学由此萌芽，照亮蒙昧世界。万物之理，数为基础，几何为根，方成